Assalamualaikum. Hi. This presentation is on second part of topic 5 on the bioreactor engineering. So in this presentation, we're going to look at the scaling up of third tank bioreactor. The knowledge of scaling up is really important as a chemical engineer or a biochemical engineer, because the product that we produce, it will be commercialized. It will be sold to the community. So we need to know how to increase the production volume from the small scale or lab scale into a full scale of commercial. The question to ponder: why would the performance of a fungal culture making an antibiotic be so different at 10,000 liter than at 10 liter? The answer is, it is difficult to maintain the homogeneity in large systems, changes in surface to volume ratios, and changes in cultures themselves due to the increased length of culture time. And we need to understand the problem in scale up, in which we need to describe what traditional culture vessels are being used and how they are being operated. Now, these are the stages involved in scale-up processes, particularly if we plan to produce a new product that we are unsure. How would the cell behave? And we have to start from the shake flask. In the fermentation or the bioreaction fermentation, we need to start with the shake flask, and this shake flask study is usually being done in the lab scale. So when we have this shake flask, then we know that we are in the lab scale stage. And next, the pilot scale, in which we increase the volume from the lab scale into a larger volume, which is known as a pilot scale. And then we succeed in the pilot scale. If we succeed in the pilot scale, then we shall go to the larger scale for the commercialization. Alright, the definition of scaling up means that you have to increase the scale of the volume of the fermentation from the shake flask to the lab scale, or from the pilot scale to the larger scale for the production. Increase in the scale, it means that you have to increase in the volume, and the problems of process scale are due to the different ways in which process parameters are affected by the size of the units itself. To increase in scale may result in a change of environment on the organism, the microbes. The parameters that should be considered in scaling up are the nutrient availability, the pH, the temperature, the shear conditions, the dissolved oxygens, and carbon dioxide concentration, if it is for the aerobic bacteria, aerobic fermentation, and the foam production. And all the above are affected by agitation and aeration, because of, we know that the nutrient, the pH, the temperature, and the shear conditions is based on the mixing, the agitation, the dissolved oxygen, the carbon dioxide concentrations in the fermentation, and also the foaming produced is because of the flow of the air or oxygen in the fermentation. This picture shows the scale up reactor. We know that from in the microbiology area, we start with the plate study, the culture plate study. We have this bacteria on the plate. Then we have this shake flask. Shake flask. And then we have this lab scale. Shape flask is still in the lab scale, but in the lab scale we also have a bioreactor, but in a small volume. Okay, this is small volume of bioreactor in the lab scale. And then we can have a pilot scale, a larger volume, okay, compared to the lab scale. And then later we can have the larger scale, the commercialization scale, okay, of reactor, in order to produce our product in the large amount to be marketed. Okay, you can refer to the textbook, the Schuler and Cargie textbook on page 288 to 290, okay, on the basic design in the stirred tank bioreactor. So here you can see that the rule of thumb, okay, in order to design the stirred tank bioreactor is the ratio of height to diameter. It is between 2 to 1 to 3 to 1, and commonly we use 2 to 1. Okay, and in the bioreactor it need to be fitted with a buffers. Okay, fitted with the buffers, and the buffer required is based on the volume, the size of the vessel, the tank. Okay, if you have a tank which is less than 3 meter, then you require 4 buffers, and if you have larger than 3 meter, then you need 6 to 8 buffers. Okay, so the width of the buffer, it can be designed, okay, by using the ratio of dt, diameter of the tank, over 10, okay, and dt over 12. Okay, so the 75% of the total vessel volume is filled with the liquid, and remaining 25% it is for gas space. Okay, which means here, if you have 1,000 liter of reactor, okay, you have to allocate 25% from that 1,000 liter for the gas space, and remaining is for you to fill the liquid. Okay, if foaming take place, okay, there is a chance of immediate contamination. That's why in the fermentation we have to avoid foaming. Okay, and there in the reactor, we always install the foam detector sensor or detector, or also known as probe, okay, in order to detect the formation of foam in the fermenter. Okay, if the height is equivalent to the diameter, okay, one agitator only required to be installed. Okay, but if the height is equal to 2 times of diameter, okay, or more, then additional set of agitators should be added. Okay, this agitator install, okay, it will be based on the volume of the bioreactor itself. Okay, the volume of the liquid that will be filled in the vessel. Okay, so proper spacing between impellers, okay, impeller to impeller, should be the diameter of impeller is less than the height of impeller, which is less than 2 times diameter of impeller. Okay, you need to have the spacing between impeller to impeller also. The installation of multiple sets of impellers, okay, it will improve the mixing and mass transfer in the fermenter. Alright, this is the standard geometrical dimension of a stirred tank bioreactor. Okay, for example here we have 3 impellers with 6 blades. Okay, impeller, of each impeller it have 6 blades. Okay, and we have 4 buffers, right? So the dimension, in order to design this dimension, can use the ratio that we mentioned before, which is height to diameter, whether 2 to 1 or 3 to 1. Okay, but mostly we use 2 to 1. Alright, so each of the ratio, okay, based on the rule of thumb, okay, you have to remember. Right? Okay. The data required for bioreactor selection and design are, we need to know the type of species of the microbes that used, and we need to know the growth and oxygen requirement, whether it is aerobic or anaerobic. Okay. And on the shear and rheology effect, okay, because we're having this agitation and mixing there. Alright, and the sterility, this is very important in the bioprocess or biofermentation. On the hygiene, okay, we need to know the procedure how to sterile, okay, by using steam blasting, right? Or in the lab, we are using autoclave, okay. And then we need to know the volumetric output, okay, the foam formation, okay, based on the previous study. Okay, we have to do homework whether there will be a foam formation during the fermentation, so that later when we have this production, we can take the precaution. Okay, and we need to know the final product, okay, of our fermentation. Okay, the cleaning and sterilization, again on the sterility, the heating and cooling system, okay, because of the temperature required. The materials of construction, yes, this is being learned in the material engineering. Okay, whether to use the stainless steel, the carbon steel, alright. And then the measurement system. Okay, talking about the materials, usually for the fermenter, we are using the stainless steel. And you know that there are different coat of stainless steel. Okay, and widely used is 316. Okay.